So now we travel back to Europe. This time we come to Sweden. And Zoe Fisher is going to tell us a little bit about another comparison, like earlier with kinetics and time resolve, now this time with neutron and X-rays for protein crystallography. So good luck. If you can press the start here, just show you.
It's going. Good. Thank you so much. And thank you to the organizers for the invitation. I'm very happy that there is at least one neutron talk in these few days. So yes, so I'm very grateful for the opportunity. I am Zoe Fisher. I work at the European Spallation Source, which is the newest neutron facility currently under construction in the south of Sweden. And I'm also affiliated with Lund University. So I guess I'm sort of representing both institutes. And I will tell you a little bit about using neutrons and how it can be used for structural enzymology, drug binding studies. So it is truly a complementary or other method or way to characterize your protein crystal. The goal for X-rays and neutrons is essentially the same. There's no mystery here. It is single crystal Bragg diffraction. And the goal here is to go from a crystal, do some diffraction experiments, determine your structure or solve it, however you wish to put it, and hopefully finally learn something about how these systems may work. With neutrons, there are a few nuances or differences from the normal workflow you would do with single crystal X-rays, and that really comes to the sample preparation. I will spend a few slides explaining why we need big crystals, how we make them, and also the importance of isotope labeling. And of course, the actual probe, the neutron beam in this case, can come in a variety of flavors. Unlike the standard synchrotron experiment where you have a single monochromatic X-ray beam, with neutrons, depending on which facility you visit, you have choices. So you can have monochromatic neutrons, quasi-Laue, or at spallation source, you may have time of flight Laue. And these impact how you do your data collection strategy, but ultimately it doesn't affect what you get at the end, which is an MTZ file. And then we do structure refinement and modeling, very similar to how you would do for an X-ray structure. We even use the same software suites. So X-rays versus neutron scattering magnitude is quite different conceptually. So I would take a slide or two to just drive this point home. So when you look at nuclear scattering length density maps, they are conceptually and in practice very different from electron density maps. And this really has to do with the scattering magnitude of the two different probes. In the dashed line here in this graph, you can see a very nice linear relationship between the magnitude of the X-ray form factor and the number of electrons, which is of course the atomic Z number. And there's a very nice linear relationship between these two things, which means even if you don't know what you have, just based on the scattering magnitude, you can figure out how many electrons this thing may have. Neutrons, however, is this extremely erratic, random appearing distribution of neutron scattering lengths. And all of these had to be determined empirically because there is no way to predict how this nuclear interaction would work for a specific element. Neutrons also actually scatter off the atomic nucleus, not from this cloud of electrons. So it's a point scatterer in this concept. And very powerfully for biologists is at the very low end here, where you have hydrogen at the very beginning of the periodic table with one sad little electron. You, in theory and practice, get almost no scattering information from such elements in an X-ray experiment. But with neutrons, you can see protium and its isotope deuterium have very large and very different scattering lengths. And this becomes then the real reason why many of us have chosen to include neutron protein crystallography in our enzymology studies. It is really this ability to get information on the hydrogens. So H atoms do have special neutron scattering properties. If you have an idle moment, I recommend to go to this NIST website and check out this beautiful interactive periodic table where they have in a very detailed way populated each element with all the neutron scattering length data and cross sections that were empirically measured. So you can click on a particular element, it will open this box at the bottom, and here you can see a very nice summary of the scattering properties of the isotopes of the same element. So I'm showing here hydrogen. First off, we can see that it has a very large incoherent cross section, which is bad news. Your protein is full of hydrogen. Roughly 50% of atoms by count in a protein are hydrogens. And when you have this large of an incoherent cross section and not a very large coherent scattering length, this can really cause problems in your experiment. But the isotope directly below deuterium have a much lower cross-section and actually a very strong positive scattering signal. So simply by replacing as many hydrogens with the isotope deuterium, you can really dramatically improve your experimental data. So hydrogens are in fact everywhere. They're not a nuisance in X-rays. In fact, we tend to ignore them or worse, guess where they are. But with neutrons, you can't get away with this. They contribute in a very real and significant way to your data. So we have to deuterate, and that means we label with deuterium, and it is essential. You have to do some deuteration to some extent in order to get measurable data. And our Japanese colleagues published a paper where they had a beautiful figure that illustrates exactly the power of deuteration. So on the left with the pink map, you see a HD exchanged crystal. And what you immediately notice as these unexchanged hydrogens covalently attached to these carbons in a phenylalanine actually lead to something called signal cancellation. They have a negative scattering length. They actually cancel some of the positive contribution from the adjacent carbon. So while this map looks pretty ugly if you're an X-ray crystallographer to neutrons, this is normal. It's perfectly normal to see these kinds of maps for HD exchanged crystals. If you have a ton of money, like a wheelbarrow full, you can deuterate or fully deuterate your protein, and then you get these very clear, beautiful, very easy to interpret maps. But it does come with a cost associated with it. Typically, your yields go down when you grow these things in bacteria or yeast. And we also know that deuteration causes some very subtle but real biophysical changes to the properties of your protein, which can interfere or complicate crystallization. So that also requires then perhaps some additional optimization and fine screening. So up to now, more than 2/3 of deposited structures in the PDB from neutron crystallography are in fact HD exchanged. You need far less deuterium to achieve this, and you still get deuteriums in the places you want them, which are usually waters, histidines, things you're interested in for enzymatic reasons. And I very much appreciated Gerard's talk yesterday, where he emphasized the need for people spending a little more effort and time to grow nice, large crystals. In neutrons, I would say this is what we spend the bulk of our time doing. Once you have a good enough crystal, the downstream things tend to work. But if you don't have a big enough or good enough or well enough labeled crystal, the downstream experiment will surely not give great data. So we do spend a lot of time growing large single crystals. And by that, we mean it needs to be roughly 1 millimeter cubed, which is big enough where you do not need a microscope to see them or mount them. There's no magic involved. We use really the methods that were standard in the 70s and 80s for X-rays before X-ray sources became so bright. Back in the day, people needed big crystals for X-ray crystallography. And we use very much of the same approaches. And vapor diffusion is the most common, but there's also examples of batch dialysis and counter diffusion capillary crystallization. And then we modify some parameters to really just encourage a single crystal to grow as big as it can. We do use temperature cycling. pH often has to be optimized, especially if your protein has been deuterated. And we also do tricks like microseeding, and I'm personally quite fond of crystal feeding. The take home here is that big crystals require a lot of protein. And we tend to grow the biggest crystals in these very large 9-well glass plates. It's something you buy from Hampton Research. It's called a sandwich box. Your reservoir is 25 mils, and your individual crystallization drops can be anywhere from 50 up to 1,000 microliters. So yes, a lot of protein, but well worth the effort. And you always get very nice X-ray data from these crystals as well. So once you've labeled your protein and you have a nice big crystal, now it's time to try to find a beamline. There are not very many in the world. I summarized them here. There are currently 5 instruments available to the user community at spallation or reactor sources. There are no home sources for neutrons. You cannot have one of these like you may have an X-ray machine. So you have to travel to a large facility. In Europe, we have the Ladi and Dali instruments at the ILL. We had BioDiff at FRM2 in Munich, but they have not had neutrons for 3 years due to some reactor problem. So BioDiff, we hope, will come back maybe later this year or next year. And Imagine at Oak Ridge National Lab in Tennessee. In Japan, we have IBIX, and again at Oak Ridge, but at SNS, we have MANDI. These 2 beamlines are built at spallation neutron sources, which is a another way to produce neutrons. We are currently building the NMX beamline at the ESS. I will have a few slides about that later. And where I worked before was at Los Alamos National Lab, where we had the protein crystallography station at the Lujan Neutron Scattering Center. And this beautiful detector is shown here at the bottom that we had. And the idea here is to give you a sense that none of these beamlines look like each other. They are all constructed in a very interesting and weird and wonderful way. They all have some cobbled together software to allow data collection and processing, and it is unique to each and every instrument. The reactor sources tend to use this circular drum that has a scintillation film with a very fixed geometry. So inside this drum is where your crystal goes. So you are quite limited in the unit cell. You can resolve maybe up to maximum 100 angstrom on any edge. Whereas IBIX, MANDI, and the PCS, we had more flexibility in the detector arrangement and allowed you to go to slightly bigger unit cells, but still some limitations. And none of the technology of these detectors are remotely the same. We have a helium-3 filled gas balloon, we have anger cameras, and we have this scintillating film. Ultimately, however, the data you get out of each of these is an MTZ file you can take home and refine, and I have not really noticed a difference between the data sets collected at these different places. We like to compare neutron source as a candle and synchrotrons like the sun. This is the flux difference we are talking about. It is many orders, 10 orders of magnitude, roughly difference in the flux. And this really impacts the throughput. The measurements tend to be very, very long. The shortest you can practically do is maybe a week, up to 4 weeks of data collection time. Because of this time, also these measurements tend to happen at room temperature. However, unlike X-rays, we don't have radiation damage, so the crystals are happily sitting in the beam if they're stable by themselves. So that is not a problem. It just is not exactly a fast experiment, which means that there is very little pressure to actually design any kind of automation. You tend to rotate your crystal by a few degrees every day or maybe twice a day. So there is, yes, so it's still quite manual to do the data collection. We can overcome this low flux to some extent by growing large crystals and deuterating them. You can maybe bring down the time by half or something like this by growing the biggest crystal you can. And you can see here the number of deposited structures. To date is only 232 deposited neutron structures. So very, very, very few. Of these, maybe 50 or so represent unique protein neutron crystal structures. The rest are mutants or the same thing collected at another pH or with the ligand soaked and so on. And ultimately, because of the time required to collect these things, the data really suffer from low completeness. We never ever are awarded enough beam time to collect a proper full complete data set. And this is really an issue in the field. Also, the flux and time problem leads to low I/sigmas because of the low completeness and the rush to get as much data as you can in 4 weeks. You also often end up with fairly low redundancy. So these are issues to keep in mind. It's not that we wouldn't like better data, it's that there is not enough time to do it. And you hardly ever have a chance to repeat anything. You may get one chance for one particular mutant or one particular structure, and then you have to do the best you can with the data you do have. Data refinement has certainly evolved since I was a postdoc. When I was at Los Alamos, we had an in-house version of a neutron plug-in for CNS that was incredibly powerful and stable, but it is not maintained anymore. So there are still a few copies floating out there, but it is not generally widely available. You could also cobble together and do something in shell X by giving it neutron scattering length lookup tables, but that didn't allow for joint refinement. So only you could use shell X if you had a very good neutron data set by itself. But today, Phoenix has really taken over, and the joint refine mode that is in the previous release of Phoenix is what many or most people have used the last decade, I would say. So you typically do your X-ray refinement until your R factors converge. That includes placing all your ligands, your water molecules, everything you possibly can. You then modify your model by introducing hydrogens and or deuteriums in the appropriate positions by using ready set. And then you go for a joint refine mode. This has changed now in the newer releases of Phoenix. So I hold on to this older one because I like how it works. And here you essentially have a single model with all the hydrogens, deuteriums placed. And then you refine simultaneously against your X-ray and your neutron data set, both ideally collected at room temperature, ideally on even the same crystal. In practice, this is a bit difficult to do. Of course, you would want to collect your neutron data first and then fry it with X-rays at room temperature. But so in practice, this is difficult to achieve. So this all works fairly smoothly. The refinement works fine. However, there's a lot of manual adjustment and intervention required from the crystallographer. And Poot and Phoenix are generally okay placing the protein itself, but as soon as it comes to modeling hydrogen bonds or water molecules that have to be oriented in specific ways, they are not very good at all. And also Poot is not good at real space refining deuterated amino acid side chains. The things atoms just tend to fly away. So there are some issues there. Perhaps a fix to the monomer library for Poot would be helpful. Yeah, so that is an issue. So we do spend most of our time doing manual inspection and adjustment, which is extremely tedious since you now have double the amount of atoms. So waters are not just oxygen anymore, they are now ×3 the number of atoms to take into account. So that takes a lot of time. Anyway, so why do we bother doing any of this? Well, you can guess the secret is that we really want to see hydrogens in our structures. Half the atoms in your protein are hydrogen atoms. They're integral parts that drive protein folding and enzyme mechanisms. They also, of course, tell us whether an amino acid side chain is charged or neutral. They participate in hydrogen bonding, and many enzymes, as shown here, actually use proton transfer as a key component. And you will not see a proton in an X-ray crystal structure, but in neutrons, you can trap them. So we know these things are important, we know they're there, but we rarely get explicit experimental information on them. So neutrons can really help us here. So here I just show some images of what it can look like when you have a nice neutron structure of, for example, a hydrogen bonded water network in a protein active site. Neutrons can help us distinguish catalytic solvent species. For example, you can easily tell the difference between a hydroxide and a hydronium, for example, just by the shape of the density. With X-rays, just to drive this point home, you will just see the same oxygen. So this is really, really key if you have a catalytic water, for example, in your enzyme mechanism. And beautifully, you can really distinguish between lysines that are charged in neutral, and the same for histidine side chains, just as an example. And this extends to, of course, the whole protein. And these can really be key points if you're trying to understand a particular mechanism. Even so, for drug or ligand binding studies, even more so, I would say, and I think in the future, neutrons will be more and more useful for drug design and inform medicinal chemistry, for example, because we get very specific information on solvent displacement, hydrogen bonding disruption when a ligand binds, and we can understand which tautomer is bound and what is the protonation or deprotonation state of a drug or a molecule bound to its clinical target. And not only for drug design synthesis, but also for computational methods, this information, experimental information from this, without guessing where things are, you actually can pinpoint where things are under specific temperature and pH conditions in your crystal. So here below, again, is just a smattering of examples from the literature on HIV protease, xylose isomerase, and beautifully you can see here the exchange deuteriums of the substrate of xylose isomerase are really much more clearly defined in the green neutron map as opposed to the red electron density. So neutrons are used to advance our knowledge for drug inhibitor binding. They're very useful for redox proteins. They don't cause changes in the metal coordination because they, again, don't cause any of this ionizing radiation effects. And they're a useful tool for general enzyme mechanisms. And you really, we joke as the facility people that neutrons are really the last resort. If you've made all the mutants, you've done 100 X-ray structures, and you still are unable to infer or guess if a particular hydrogen bond exists or where a catalytic water is, neutrons really are the answer. It is really the one way to obtain these kind of answers. Yes. However, the user community is small, and we're always trying to attract new users, and this really rests on the facilities. We have to ease these bottlenecks for users. We need to help them grow large crystals. We also need to help them with deuterium labeling, not just the cost, but there's also some technical expertise involved with doing delabeling. And users need access to equivalent room temperature X-ray structures, which are not super common for single crystal methods at synchrotrons these days. I seem to, wait, is it coming back? Let's wait a second. Anyway, and of course, once you have gotten through these bottlenecks and have a data set, unless you are fairly seasoned, you will struggle to interpret these maps as well. So shown here is a beautiful example of signal cancellation, where if this was your electron density, you would say, wow, this map is crap. But if you know it was a HD exchange crystal, this is absolutely to be expected. So at least initially, it's a good idea to help users work through their actual experimental data as well, to help interpret and place hydrogen atoms and satisfy the hydrogen bonds that are there. So the ESS, like I said, is being constructed in Lund. The facility is soon complete. When we are done, we will be the world's brightest pulsed neutron source. It is driven by a proton accelerator that is currently spec'd to operate at 2 megawatt power. We will use a tungsten target and advanced moderator system to produce the highest neutron flux possible. We have 3 experimental halls, and today there are 15 state-of-the-art beamlines under construction, and they're in various stages of completion. We aim to have first neutrons by the end of this year, so that's very exciting. We will welcome some friendly users and close collaborators, hopefully next year, and we aim to launch the full user program the year after. One of the 15 beamlines currently under construction is the Neutron macromolecular crystallography beamline called NMX. It is a state-of-the-art time-of-flight Lowy instrument, and it is not just a schematic on a slide anymore. Actually, we have many of the technical components on site and installed even. So most of the 150-meter neutron guide is installed and even covered with white shielding. You can see here. Back here is actually the target bunker. So the neutrons are produced here. They are extracted through 2 experimental holes. The beam is so long, all the way out into the E buildings where the cave is finished. So we have the experimental cave, control hutch, and quite a generous sample preparation lab where we will help users grow large crystals and mount them for data collection. We have a chopper system in place. Detector is in a prototype stage and still undergoing some testing. But when that is finished, we aim to have 3 detectors on robot arms. So we won't be constrained by this fixed instrument geometry that exists at other beamlines. The idea is that we can move the detectors to get optimal reciprocal space coverage, and we can also move them back to accommodate larger unit cells. So couple this advance with very, very high flux, we really hope to really push the envelope forward in the coming years. In addition to beamlines, ESS is also invested quite heavily in labs. We have tons of lab space on site, chemical labs, all-purpose labs, and also a highly specialized team that I am a part of, where we really help the neutron user community with access to deuterated materials and protein crystals. We offer chemical deuteration of small molecules of huge range. We have a 15-page catalog of things we can synthesize, deuterated, for small angle scattering, reflectometry, crystallography, whatever your heart desires. If it's synthesizable, we can probably deuterate it. The same for biodeuteration. We deuterate things in E. coli, algae, and yeast, mostly for recombinant soluble protein production, but we also get asked for other things like deuterated DNA or lipids, phospholipids from yeast, and so on. And then finally, we have a crystallization lab where our sole goal is to find, screen in large volumes to really optimize protein crystals. And we are part of a bag, so we have regular access for room temperature X-ray screening of these crystals. So some final reflections. NPX has really come a long way in the last decades. Progress is slow, and there is some room for improvement. We do need more beamlines. Biodiff in Germany being down at the moment is a huge hit, and we see it in the statistics as well. Since they were not in operations the last years, there is really a decrease in the number of new structures being determined. So we need biodiff back, and we need to hurry up and finish NMX. There are also some improvements required, I think, to make the software more easy to use and more intuitive, and perhaps some efforts into making hydrogen atom and hydrogen bond modeling better or more automated. However, we really do need the ESS and this high flux to enable faster data collection and smaller samples. And our goal is an order of magnitude smaller crystals and a structure in 1 or 2 days. So then imagine you had 3 weeks of beam time, you can maybe collect 20 structures instead of just 1. So I think systematic enzymology studies and or doing more proper drug binding studies, for example, with multiple compounds and not just one, would be possible. And of course, we integrate this with support from our support laboratories. So the burden is not just on the users. So thank you for your attention, and thanks to amazing colleagues and a team. It's so good. We have a very young and dynamic NMX instrument team at the ESS, so I really think things are looking very good for neutrons in the next decades. Thank you very much.
Thank you, Zoe. Really, really interesting. I'll start by throwing one or two questions related. So you said that you scared us, saying crystals have to grow very big. This is really hard. You need a lot of X-ray data. But can you comment on the speed that it takes currently ILL and the speed it will take for a data set at the spallation source, but also comment the interest of using Laue that you collect more and less. And also what I'm also interested is the space group. If I have a high symmetry space group, it should be faster to collect a complete data set.
For sure.
Do I need a crystal as big, for example?
The size is hard to get away from. It's not just the symmetry or the labeling. It really is the flux. So you really need as many neutrons on your crystal as you possibly can get. And the beams are not easy to focus either. So the beams tend to be quite large. So you need a sample to match the beam size. That's part of the problem. High symmetry is, of course, fantastic. However, in practice, out of the 232 deposited structures, most tend to be P2, P21, a few orthorhombic, C2. We have very few examples of high symmetry groups. And I don't know why, if there's some inherent limit in the size achievable, or if those proteins have just not been tried. So that is hard to say. Laue, yes, of course, the idea is that if you have Laue neutrons, you need far fewer crystal settings, so you can be much more efficient in your data collection. But again, it's a flux game. So we had a full Laue instrument at Los Alamos, but we had so few neutrons, 10 to the 6 per second per centimeter squared. So, I mean, that is like, I don't know, home sources were probably better than that in the 70s, I guess. Yeah. So, even with time of flight Laue, it still took weeks to collect a data set. So we really need, I think the flux is the key. Yes.
And in the Laue side, how is the software? Because someone recently collected X-ray data with Laue data and went to look at CC4, and the program had not been updated for 15 years or something. So it didn't work with the current IGA data, and it was a pain to deal with.
I imagine so, yeah. For the Laue data processing at Los Alamos, we used the old Darsbury suite called L-Scale and LaueGen from John Helliwell's group. So that had been cobbled together and adjusted to work on that particular beamline. It's a typical problem where it only worked there. And that software now, nobody has been maintaining that for some years. At Oak Ridge, the other spallation source that does TOF Laue, they use Mantid and other things for their data reduction. So I think at ESS, we have a team of people working in the data management software center that are working on tools to do this.
That makes sense.
But yes, that is a way to reduce the data collection time.
Do we have a question here?
And
Giles is gaining time of flight Laue, and David.
yes, David is here somewhere. He was here. Yes. Yes.
Hi, Alan Orville, Diamond Light Source. I'm not an expert in neutron studies, but I heard a talk recently from Dean Miles at Oak Ridge, and he was pretty high and pretty convincing on dynamic nuclear polarization as a way to enhance the signal. Is this something you're considering in Lund as well?
We are not at the moment. It is very interesting. I visited Dean at Oak Ridge 2 years ago, and he showed me the setup they were putting on Imagine at the HIFER instrument. So they are testing this, and the initial measurements they've done, it seems to work pretty good. This would, of course, negate the need to deuterate your sample, which is a major pain and a cost. So if there is a way to polarize the crystal and get good data, but this has to be demonstrated for something more than just lysozyme. So I know Dean is working on it, and they have a perfect test bed at Oak Ridge because they have this extra beamline that they can test things with. I don't think we have this luxury in Sweden. We're under enormous pressure to get this beamline up and running. Of course, with flux and deuteration, should just be fine. But indeed, we have actually a large team of scientists at ESS working on polarization for the other beamlines, including some diffraction, small molecule stuff. And I think we will gain a lot of knowledge in the first years of operation, how that is working out. So we do have the technical expertise at ESS. We are just not yet considering integrating it into NMX, but not to exclude it.
Any more questions? Are there questions on Slack?
I was really interested by the crystal feeding approach that you described. Could you briefly explain how it works?
Yes, so usually you start with sort of a normal vapor diffusion sitting drop, and once you have a reasonable crystal, we often move it to a fresh drop that has been pre-equilibrated. And then you feed a little bit of a few microliters of protein solution every 3 to 4 days. Very slowly. You don't want to dissolve your precipitant too much, but you don't want to add extra salt to induce new nucleation. So this is a very careful dance you play by feeding small amounts of fresh protein to that drop once or twice a week, and then monitor the growth. Not all protein crystals will do that. This has worked really well for about 3 unique protein projects we did in the last years. There is a paper, if I have the reference for it, a 2016 paper we published on exactly this, where there is like really nice details on this paper at the bottom, Manzoni et al. Acta D from 2016. In there is beautiful description of feeding versus macro seeding, and that is the text, that is the example we use, not just for galectin, but for other projects as well. So if you're keen on feeding your crystals, this is a good guide how to go about it. Yes.
Thank you.
Right, I think there are a few questions Slack, but we can answer them there. I think for interest of time, let's thank Zoe once more, please.